Good morning, sir. This is my answer in fish genetics exam. Number 1. Define genetics and its importance in fisheries. Genetics can help to improve fisheries management. It is very important in fisheries in terms of breeding program. Efficient breeding program will be crucial to develop not only to reach the production's goal, but also to reduce production costs, improve disease resistance, improve utilizations of feed resources, and improving product quality. The history of animal breeding shows that the principles of selections have been applied for thousands of years. Long before the theoretical background was developed with considerable success. The main purpose of breeding program is to develop animals which more efficient utilize available feed, land, and water resources. It is very probable that pursuing the goal we will also select animals which are better adapted to the captive environment as they must produce well under these conditions. Number 2 questions. What is the chromosomal and chemical basis of heredity? Chromosomal is a structure found inside the nucleus of a cell. A chromosome is made of protein and DNA organized in genes. Each cell normally contains 23 pairs of chromosomes, while DNA it is known as the chemical basis of heredity. DNA is made up of combinations of nitrogenous bases, sugar molecules, and phosphates groups which are linked by different bonds in series of sequence. Number 3 questions. Explain the concept of population genetics. Population genetics is the branch of genetics that conducts the study of the genetic compositions of populations. It combines genetics, evolutions, natural selections, selective breeding, statistics, and mathematics. It makes computer and mathematical models and conduct field research to test the models. While populations genetics spends their time doing two things: describe the genetic structures of populations and theorizing about the evolutionary forces that affect populations. Number 4 questions. What is the importance of selections in fish stocks management? The uncertainty is the parameter estimated can be incorporated in the stocks assessments to produce a variable results under a given management scheme. This variability provides flexibility to managers, but also has implications for the stocks depending upon where in the range of possibilities of the managers decide. Number 5 questions. Differentiate hybrids from inbreeds and plant a good crossbreeding program. So, hybridization is the process of crossbreeding genetically different individuals to produce offspring, whereas inbreeding in the crossing of two closely related parents, close relatives who share very similar alleles. Inbreeding is more likely to cause genetic abnormalities, unlike hybridizations. 6 questions. Identify biotechnologies in fish genetics and determine how they could help increase productivity. Biotechnology allows scientists to identify and combine traits in fish and shellfish to increase productivity and improve quality. Scientists are investigating genes that will increase the productions of natural fish growth factors as well the natural defense compounds marine organisms use to fight microbial infections. Number 7 questions. Mitosis and mesosis. Different mesosis from mesosis. Ito po yung sagot ko. Mitosis is the process of non-nuclear divisions of eukaryotic cells that occurs when the parent cells divides it to produce two identical daughter cells. Mitosis is conventionally divided into 5 stages known as the prophase, prometaphase, metaphase, anaphase, and telophase. While meiosis is the process where single cells divides twice to produce 4 cells containing half the original amount of the genetic information. These cells are our cells, sex cells, sperm in males, eggs in females. During meiosis, one cells divide twice to form daughter pram cells. Differentiate DNA from RNA. DNA is a double-stranded molecules, while RNA is a single-stranded molecules. DNA is stable under alkaline conditions, while RNA is not as stable. DNA and RNA base pairing is slightly different since DNA used the bases adenine, thymine, cytosine, and guanine. RNA uses adenine, uracil, cytosine, and guanine. Number 9 question. Discuss how and why DNA replicates. Replication occurs the 3 major steps: the opening of the double helix and separations of DNA strands, the priming of the template strand, and the assembly by the new DNA segment. During separations, the 2 strands of the DNA double helix uncoil at a specific location called the origin. While DNA replicates because whenever a cell divides, the 2 new daughter cells must contain the same genetic informations or DNA as the parent, or the DNA as the parent cells. Explain the importance of transcriptions and translations. The goal of the transcriptions is to make an RNA copy of the gene's DNA sequence. For protein coding genes, the RNA copy or transcript carries the information needed to build polypeptide proteins or protein subunit. Eukaryotic transcript, you need to go through some process steps before translation into proteins. Translations enables effective communications between people around the world. Its carrier from the transmissions to know a protector of the cultural heritage and essentially to the development of a global economy. Highly skilled translator as the key. Number 11 question. Discuss how proteins and amino acids are synthesized. During translations, ribosomal subunits assembles together like a sandwich or the strand of mRNA, where they proceed to attract tRNA molecules tethered to amino acids or circles. As long chains of amino acids emerge as the ribosomes decode stage mRNA sequence into a polypeptide or a new protein. Number 12 questions. Explain the principles of YY mail technology and the process of how to produce. YY males produce a converting females into a male populations. Yun lang po yung simpleng sagot sa number 12. Number 13. Explain the different kinds of artificial selections method, mass selections, family selections, and multi-trait selections. Mass selections. These kinds of artificial selection, which the simplest form is known as individual selections. In mass selections, the best individuals are selected from the populations for breeding, and remaining the individual is discarded. The progeny of the selected individuals are usually better on average than the original populations. While family selections is used when the heritability is small and there are uncontrollable source of environmental variance. It involves the choosing entire families using groups of full sieves or half sieves. Families are selected on the basis of their mean phenotypic value, while multi-trait selections, the best approach for selecting an animals considering multiple traits is the use of the economic selection index. The overall breeding goals is to improve multiple economically relevant traits to selecting animal using the selection index. Number 14 questions. Advantages of crossbreeding. Crossbreeding is used to produce superior fish for growth out production fish, while selection is used to create superior food fish. One of the most common breeding program is aquaculture is productions of sex reverse brood stocks produce monosex populations for growth out.